So this karyokinesis, nuclear division, the nucleus start dividing. Once the nucleus division happens, cytoplasm start dividing. So when you see that this nucleus is divided, then cytoplasm start divide, making two new cells. Which is cytoplasm division we called cytokinesis. Karyokinesis is a nuclear division. That is very important. For example, they have a different stages of that mitosis. For example, you have to remember, I don't think I have enough space over here. I will write it here. Mitosis M. Remember, this is the stages. PMAT is a prophase, remember, this is the stages of mitosis: prophase, metaphase, anaphase, and telophase. Prophase, every DNA start packing inside the nucleus. This is I'm just drawing the nucleus. The DNA start condensing. It's just packing everything. You're going to see start visible this DNA. M phase, everything start line up in the middle. Remember, F is packing, M is what? Okay, everything in the middle. Anaphase, the fibers forming here. This chromatid start moving apart from the middle. Okay, it's going from here. It's going away from the midpoint. It's going towards that two different poles. Then telophase. So in the cell, you're going to see two different nucleus because it's already going two different poles. It start dividing. So you're going to see two. Remember two nucleus in that same cell you're going to see. This is a nuclear division. You remember cytoplasm still intact. It doesn't divide it. Then after this, this cytoplasm start cleavage furrow is forming and start dividing. It's become two new cells. Then make sure you know that what is that mitosis, what are different stages, how you can remember these things. So make sure you remember. Remember PMAT. Remember, some people take a PCAT for pharmacy entrance exam, MCAT for medical school. So remember this PCAT. P is for phase, M is for metaphase, A is for anaphase, T is for telophase. Each function wise also you can remember this. Remember the first letter. Pro is for what? Packing. M is for in the middle, everything in the middle. Anaphase is away from the midpoint, going to that pole, away. T is for two nucleus. Then cytokinesis is this portion, cytoplasm dividing. Now remember that cell cycle control. This cell cycle is very controlled, regulated mechanism in our system. So the regulators, some signaling molecules can regulate So one of the things, if your cells some mutation, think about is a mutation. You're going outside, exposed to UV often. Your cell is mutated. The mutated cells, you don't want the cells to go through that cell cycle and divide and making that mutated cells become more and more. So instead of that, your cells will let that regulate and going for that apoptosis. Apoptosis is a programmed cell death. Telling the cells you have something issue, you don't want that to divide and making the mistake again. So go through that death. Just to signal, tell the signals to remove the cell. So this is the one apoptosis is very important for that regulating these kind of cancer cells and other things in our system. In the cell cycle also there are some regulatory things in between that checkpoints. In that for example here is in between there is a checkpoints. Checkpoints are that things for example if you're going from one country to different country, so there are that checkpoints. They look at your passport, other things, make sure you are the right person. If you have everything is right, they allow to go to that next place. So there are checkpoints are very important for that going from one portion to one part of that cell cycle to another. So that if you're not capable of going from G1 to S, your system say you stay here and mature, and if you have any issues, make sure you that fix it, then go for that S phase. If you cannot fix it, then you go for that apoptosis. So if you have any dysregulation or uncontrolled growth of that cells, if any damage to that checkpoint, is that can cause what? Cancer. Programmed that the program has some issue, then start dividing cells more and more. Instead of doing this kind of division, more in faster level cells are dividing, which is called proliferation of the cell that can lead to cancer, uncontrolled cell growth. DNA structure. DNA is a deoxyribonucleic acid. Where do you see DNA? Inside the nucleus. They are double helix made from the nucleotides. They are bases, nitrogenous bases, adenine, thymine, cytosine, guanine. So A always goes with what? T. C goes with what? G. Think about here is G. You have to make this one. If I give you one pair, you have to make that what are the next parallel to that other thing. So is this one C or this A, T. Okay, how are you going to build this? G should be C. C should be what? G. A should be what? T. T should be that A. So this is the DNA strand. Okay. So that's why it's building that pairs. DNA replication, I mentioned that making the copy of the DNA. So semi-conservative method. So if you use this one, A, T, C, G, A, T, this is going to be T, A, G, C, T, A. That's another that strand. So here this one of them going to be making the copy of that one. So this one, if you're making this one, so the same thing over here. Okay, it's going to be exactly like that. So but this one A, T, A, G, C, T, A. It's going to be look exactly like that. So that's the way they're making that new copies. Each strand serve as a template. ensuring genetic continuity. You don't want something else. You want a copy of same thing. So genetic engineering and biotechnology is growing so fast these days. So we making that DNA, inserting foreign DNA inside for them, for the repairing the DNA, or maybe that some plants better yield, that pest control or surviving the certain season. All these the reason they inserting that better genes into that DNA, inserting DNA parts to that other organisms. Applications like medicine and agriculture. Agriculture is the plants, they better yield, survival purpose from that insects and stuff, or that seasons. Protein synthesis. Protein synthesis is coming, starting from that DNA. DNA has a DNA to mRNA, messenger RNA, to protein. DNA is a double stranded, we know that. Single stranded proteins are like this. From here to here, DNA to RNA, which is called transcription. This one mRNA to protein at ribosomes. Protein synthesis where is the protein forming? On the ribosomes. So these are the process, DNA to RNA to protein, which is a transcription and translation, which is a central dogma of biology. Okay, so this is a cell structure and basic structure and things. If you need more information, please let me know. Look at my other videos. We can get more information. Thank you for watching.